This presentation explains enzyme action. We're going to cover a number of the learning outcomes. So we're going to look at enzyme specificity, see what that means. In order to do that, we need to be talking about active sites. We're going to describe the role of the active site in enzyme function. And we're going to look at the lock and key model, which helps us explain how enzymes work. So here we've got our enzyme. It's a protein molecule, and on one part of the enzyme molecule in one region there will be a particular three-dimensional shape. So we've got a three-dimensional shape. This is sometimes referred to as a pocket, or more specifically as the active site. Now, different enzymes have different active sites. We've looked at salivary amylase, the enzyme produced by the salivary glands in our mouth. Well, all of the salivary amylase enzymes, all of them that are produced by the glands, they have exactly the same pocket, and that will allow them to work on only one particular substrate, and that substrate is starch. This is the lock and key diagram. This helps to explain how enzymes work. So it's an analogy. Here's the key, and we're saying the key is our substrate molecule. So there's our substrate molecule. Now that substrate will only fit in a particular lock. It will only fit in one lock. And obviously the lock is the enzyme. Here's the enzyme here. And we can see that substrate will fit perfectly into the enzyme. The key will fit perfectly into the lock. And when it does so, we often talk about an enzyme substrate complex. So that's the lock and key model. So here's a worked example for us. We've got the enzyme here, and it's got a very particular shaped active site. It's specific. It's specific only for these two substrate molecules. It's only these two substrate molecules that will fit in. When they do, we get this enzyme substrate complex. We get the perfect match, the perfect fit, and as a result the reaction can take place. It's being catalyzed. So this is an enzyme catalyzed reaction. We started off with our two reactants, and we've ended up with one product, and the enzyme has speeded up that reaction. So in summary, you now know what we mean when we talk about specificity. We now know that the enzyme action is all because of the active site. That's where the substrate molecules fit in. So we know the role of the active site in enzyme action and how they function. And we've seen the lock and key model. It's a useful model just to look at how the enzymes and the substrates work together.